So our last speaker of the session is Ryan Haley from the Zoo Lab at Baylor.
All right. So my name is Ryan Haley, and today I'm going to talk to you about how RAB-35 coordinates the engulfment and degradation of apoptotic cell corpses. So just to give you a basic overview of the process. So initially you have a cell corpse or an apoptotic cell. It's engulfed by a macrophage or a similar cell, and essentially it forms a phagosome around this. And this newly formed phagosome immediately begins to mature. So you have incorporation of lysosomal enzymes and gradual acidification. And this is basically aided by the fusion of early endosomes and lysosomes to the phagosome. And eventually, once you get these lysosomes fusing to the phagosome, you form something known as a phagolysosome, which essentially initiates digestion. And you actually see a shrinking of the phagosome as the cell corpse is degraded. Now, key to this process of apoptotic cell clearance are small GPases known as Rabs. And they basically function by recruiting effectors. And they don't really act directly, but they rather act as managers of cellular processes. And they do this by alternating between two distinct forms: inactive RAB-GDP form and an active RAB-GTP form. And Rabs essentially separate maturation into distinct phases. So early in maturation, phagosomes are largely RAB-5 positive, but later on in maturation, there is a distinct active removal of RAB-5 and a replacement by RAB-2 and RAB-7. So Rabs essentially kind of organize maturation, and if you disrupt them, you basically disrupt the process as a whole. So we really wanted to explore these Rabs and apoptotic cell clearance a bit more. And to do this, we kind of took advantage of a phenotype that we noticed, that anytime you have a defect in apoptotic cell clearance, you usually see an increased number of cell corpses to appear, which is what we call the SED phenotype, or cell death abnormal phenotype. So what we did is we performed an RNAi screen of all of these 29 predicted Rabs and C. elegans, and then we just screened through them and tried to figure out which of these caused the SED phenotype. And in addition to the ones I mentioned earlier, RAB-2, RAB-5, and RAB-7, we noticed that RAB-35 loss-of-function mutants contain significantly more cell corpses than wild-type worms. And you can actually see this right here. So in wild-type, these cell corpses kind of look like bun-like structures. You can see them, the white arrows point to them, and in these RAB-35 loss-of-function, you can actually see some more cell corpses compared to wild-type. And the key thing is that we've seen this in any sort of scenario that we looked at. So we looked at early embryos, 1.5-fold, late embryos, which are 4-fold, and then in the adult gonad, and we see it in every case. Moreover, we kind of know that RAB-35 is engulfing cell-specific in cell clearance, because if we express RAB-35 using an engulfing cell-specific promoter, namely PSET1, we can rescue the RAB-35 phenotype. So to begin with, I kind of want to talk about whether RAB-35 is important for the engulfment of cell corpses. So to do that, we kind of look at the embryo under two different channels. The first, which I've shown you before, is this DIC channel. And essentially under it, these undigested cell corpses are very refractile, so they look bun-like, as I've shown you, as I mentioned previously. But if we want to say if there's a protein that's recruited to the phagosome, phagosomes directly surround these cell corpses. So if a phagosome that surrounds a cell corpse is recruiting a protein, it actually appears as a GFP circle, as you can see here. So if you overlay these two channels together, you can actually see which phagosomes are recruiting your protein of interest, which would be this red arrow here, and which ones are not, which would be this black arrow here. So if you do population dynamics using these markers, you actually can see general trends and act on them. And the first marker that we're using is SED1 delta C. Basically, it's a modified phagocytic receptor that serves as an excellent engulfment marker. So I don't have a lot of time explaining this, so basically what you need to know is that if the cell corpses have this GFP circle around them, we know that they're definitively engulfed, but if they're GFP minus, we know that they're definitively not engulfed. And we basically used SED1 and SED5 mutants as positive controls because SED1 and SED5 represent the two major engulfment pathways in C. elegans. So if we knock out either SED1 or SED5, we expect a very significant, a very large engulfment defect. And what we see is that there is a considerably, in the RAB-35 loss-of-function mutants, there's a significantly fewer cell corpses that are properly engulfed relative to wild type. Although it should be important to note that this effect is much more moderate compared to knocking out either SED1 or SED5. So we kind of wanted to describe this engulfment defect a little bit more. And the problem is that cell corpses can fail to be engulfed in a couple different ways. So either the engulfing cell can just fail to recognize the cell corpus as a whole and just not initiate the process to begin with, or the engulfing cell can recognize it, but fail to form a phagosome because pseudopod extension is defective. Now, unfortunately, although pseudopod extension is pretty easy to detect because you can just label the growing pseudopods, measuring defects in recognition is a bit more difficult because you have to measure the beginning of engulfment relative to another time point. So this is one of the great things about C. elegans because development is very regular, very consistent between embryos, so we decided to pick a process known as ventral enclosure as our 0 time point. And the reason why we did this is because during ventral enclosure, on the ventral surface, you have these growing epidermal cells that stretch across. And it's these same cells that are basically going to engulf our 3 cells of interest, C1, C2, and C3, which are labeled here on the cartoon for reference. So basically, we use the same engulfment marker as before, SED1 delta C, so we can measure engulfment. So shortly after ventral enclosure, C1, C2, C3 will be engulfed. We can measure that timing to determine if recognition is defective, and then we can just track the pseudopods to see if pseudopod extension is defective. And what we see is that RAB-35 mutants actually do have delays in recognition. Actually, loss of function of SED1, SED5, and RAB-35, when we tested them, all independently and significantly increased the time it took for cell corpses to being recognized. So in wild type, it takes about 12 minutes after the initiation of ventral enclosure for the process of engulfment to begin. In RAB-35, this increases to about 16 minutes. In SED1 and SED5, this increases to about 20 to 22 minutes on average, which you would expect considering that SED1 and SED5 tend to be more severe. And what we see is that the double mutants, either SED1 RAB-35 or SED5 RAB-35, have a significant population of cell corpses where it takes more than 30 minutes to be recognized. Now, the interesting thing that we noticed is that RAB-35, SED1 or SED5, none of them actually showed delays in the initial extension. Now, I should put a caveat, it's still in progress, but we've noticed that in a lot of cases, the initial extension will be proper, but there'll be a lot of retraction. So we haven't really quantified that yet, but we don't see a lot of that in the case of RAB-35 loss of function. So because RAB-35, the effect of it is parallel to both SED1 and SED5, we suspected it might be separate from both canonical engulfment pathways. So SED1 represents the SED1, SED6, SED7 pathway, whereas SED7 would represent the SED2, SED5, SED10, SED12 pathway. So we actually tested multiple components of each pathway, and when we performed epistasis analysis, we see that loss of function of RAB-35 significantly increases the number of apoptotic cell corpses that we observe. So it enhances the SED phenotype in both SED1 and SED6 backgrounds. And we also see that it's parallel to the SED2, SED5, SED10, SED12 pathway because we've tested SED5, SED12, and the partial loss of function of SED10. And we actually see that RAB-35 loss of function enhances the SED phenotype in all of them. So essentially, these results might implicate RAB-35 as a part of a novel third engulfment pathway. So next, we decided to look at degradation, see if there were any effects there. And obviously, since I'm talking about it, you can imagine we found something. So CTNS1 is a lysosome marker. More specifically, it's a lysosome-specific membrane protein. And the key point to say here is that because it's lysosome-specific, phagosomes only become GFP positive after phagolysosomal fusion has occurred. Now, the interesting thing that we found is that RAB-35 mutants have a large population of cell corpses where we see this GFP circle, meaning that phagolysosomal fusion has occurred, but they haven't been digested yet. So this suggests that there might be a defect somewhere between phagolysosomal fusion and the end of digestion. So basically, we decided to live image these strains to observe the dynamics of degradation, and we used 2 markers. The first is Moisin GFP, and that's basically our engulfment marker. So we basically use this as a 0 time point at the end of engulfment, just so we can compare embryos. In addition, we use CTNS1 MRFP, which is our lysosome marker. So first, we decided to look at the timing of recruitment, which is the time between the end of engulfment and when lysosomes are recruited to the phagosome. This is roughly normal in RAB-35 loss of function mutants, but the process of digestion as a whole, which is defined as between the end of engulfment and when the phagosome shrinks to 1/3 of its size after digestion, that is actually significantly delayed. It's delayed by about 10 minutes in RAB-35 loss of function mutants on average. So if this is relatively normal and this is significantly delayed, this suggests that the defect is again somewhere between phagolysosomal fusion and the end of digestion. So in other words, either the process of phagolysosomal fusion or the subsequent digestion is defective. And actually what we see is when we perform live imaging analysis using CTNS1, we actually see distortions and instability in the Phagolysosome and RAB-35 units. So in wild type, after you get recruitment to CTNS1, you see the expected shrinkage of the phagosome as digestion is occurring. In RAB-35, you see a lot of cases where CTNS1 will be recruited. You'll see initial shrinking at around 40 to 50 minute time point. But what you'll actually see is, in some rare cases, about 10 to 15% of cases, it will not only stall, but you'll actually see fragmentation and blebbing of this phagolysosome, which we don't see in wild type. So because there's a possible defect in phagolysosomal fusion, we wanted to see if RAB-35 was in the same pathway as either RAB2 or RAB7, which are both involved in this process. And what we found using epistasis analysis is that RAB-35 is in parallel to RAB2, but it is in the same pathway as RAB7. And when we do overexpression analysis, we actually see that overexpressing RAB7 actually partially rescues the RAB-35 phenotype, but the inverse is not true, suggesting that RAB-35 is probably upstream of RAB7. And this is actually consistent with previous research in endocytosis, as well as consistent with the fact that RAB-35 actually comes on the phagosome well before RAB7 does. So finally, we'll just briefly talk about the mechanisms that RAB-35 is using to enable apoptotic cell clearance. So RAB-35 actually joins the phagosome in two phases. The first and weaker phase occurs during engulfment. And it's basically because RAB-35 co-localizes with the plasma membrane. So therefore, you can see it basically staining the growing pseudopods. And then immediately after immediately after engulfment, you actually see a second and stronger phase right around the time of phagosome sealing. Now the question is, we didn't really know what the second phase of RAB-35 was doing when we looked at this, because it's a little bit too late for recognition, and it seems way too early to impact phagolysosomal fusion, which occurs almost 20 minutes later. So we decided to look into that more. And what we found in a piece of literature is that during this time, there is a PI45P2 to PI3P transition that is required for phagosome maturation, but the people that were doing the research had not been able to find a master regulator for the process. So we decided to look at it a bit, and we saw that RAB-35 recruitment correlates perfectly with the gain of PI3P. So we used 2x5 as our PI3P marker, and what we actually see is that this second phase of RAB-35 recruitment, the spike of RAB-35 recruitment, occurs at exactly the same time as the appearance of PI3P on the phagosome. Likewise, we actually see that the first phase of RAB-35 recruitment perfectly co-localizes with PI45P2 on the pseudopods, but immediately when you have that second phase of RAB-35, you see an immediate loss of PI45P2. So unfortunately, we're not at the point now whether we know if it's a cause or an effect, but we know it's definitely linked to that transition. So I just want to acknowledge everybody in my lab that supported me along the way. CGC, obviously, for providing strains, because they really love me to tell them that. And then NIGMS for funding. And if you have any questions, I'll be happy to answer them.
So I have a question.
Yes.
So just following up on the last point, does DAF12, the P3 phosphatase, does it affect cell engulfment in the RAB mutant?
Which?
DAF12 is the P10, is the
So class 2 and class 3,
yes.
class 2 and class 3 PI3 kinases do. Class 1, I don't think have really been explored.
Okay.
All right. Okay, thank you.
Okay, if there are no other questions, thank you to all the speakers and a really terrific session. Thank you all for coming.